Okay, so this is the phenol red broth, and for these ones, these are the lactulose, but the same results would be true for any of the different sugars, glucose, lactulose, or sucrose that we have in the lab. And so on the left-hand side, you can see what a negative results looks like. Our pH indicator, being phenol red, has not changed. It's still a red color, being a neutral pH. On the right-hand side, we see what a positive reaction looks like. Positive for acid production, being that the phenol red has changed to a yellow coloring. And positive for gas production, we can see that bubble in the Durham tube that's inverted. And so that gas that's trapped there is carbon dioxide. And that shows that this bacteria can ferment acid production and gas production with lactulose. So this is our citrate media. On the left-hand side, we can see what a negative results looks like. We have the media and it's green. On the right-hand side, the media has turned blue. This indicates that the bacteria is producing citrate. Citrate is an enzyme that can metabolize citrate. So there's citrate in the media. So because we have citrate, the enzyme, the bacteria can metabolize it and use it as a carbon source. The reason why we see the color change is because we have a pH indicator that goes from green to blue, and we have an acid being produced, acetate, and so that acetate is a byproduct of metabolizing the citrate. Okay, so here we have 4 tubes of the Sims media. On the left-hand side, this is an uninoculated, so it looks like without bacteria added, so it would be like a negative reaction. Next to it is a tube that is positive for sulfur, and so Sim stands for sulfur indole motility, and so our first reaction we can look at is whether there is blacking of the media, indicating we have reduction of sulfur. Okay. The next thing is indole production, and so if we look at the same tube here, the second tube, we see it's negative for indole production, and that's characterized by that fluid being clear. If we look at the last tube, it's positive for the indole, indicated by that red ring because we've added the indole reagent, and it has turned red because the bacteria has tryptophanase. It breaks down the tryptophan that's in the media and produces indole, and again the reagent reacts with that indole and produces a red color. Now when we look at our last 2 tubes, we can look at motility, which is the M, and so here we can see that nice clear stab line, and so that would be negative for motility, where this one we have this clouding of the media, and that indicates it's positive for motility. And so in the Sims reaction, we actually get 3 tests in 1 tube. Okay, so this is what the reactions potentially could look like for your MRVP. So on the left-hand side, I have 2 tubes for the MR reaction, and then on the right-hand side, I have 2 tubes for the VP reaction. To be honest, I've doctored these up because, as I had mentioned in class, they're not responding how they're supposed to. So this is what it might look like for the lab practical. And so here we have our negative reaction for methyl red. Honestly, it would look a little bit more like a diluted pink, but to make it very clear for the lab practical, I would have it as this kind of unchanged color from the media. Here we have a positive reaction with methyl red after we've added the drops of methyl red into it. For our VP reactions, this is our negative reaction where there's no change to the media, and here is where our positive reaction for VP is. What I've done is I've added the drops from the cuvette, so we have VP reagent A and VP reagent B. Okay, so here we have a MacConkey agar plate. The MacConkey agar plates allows gram-negative bacteria to grow on it. So if we don't have growth, and we know we've inoculated our bacteria onto the plate, it indicates we have a gram-positive. So if we were to take staph or strep or Micrococcus luteus and try to plate it onto a MacConkey agar, we would not see growth appear. Okay, those bacteria are inhibited by the crystal violet in the bile that is in the media. Now on both sides we have nice growth, so that indicates that we have a gram-negative bacteria. Furthermore, the growth is pink